All around us, there's this hidden world, right? A buzzing, bustling civilization of microbes that influences literally everything, from our own health to the entire planet. Today, we're going to pull back the curtain and unlock their secrets by cracking open their instruction manual, their genetics.
And look, this isn't just some abstract science lesson. It all starts with a huge question that affects every single one of us. You hear about superbugs on the news all the time, right? These bacteria that are resistant to antibiotics. But have you ever stopped to wonder how that happens? How does a simple little bacterium just suddenly gain the power to defeat our best medicines? It feels like it happens in the blink of an eye. Well, the answer is hiding in their DNA. And believe me, it's an incredible story of survival, of sharing, and of mind-boggling speed.
All right, so to solve this whole superbug mystery, we've got to start with the fundamentals. We need to take a look at the bacterial blueprint itself, that basic code that makes a bacterium, well, a bacterium.
Now, unlike our own DNA, which is super complex and packed into a bunch of different chromosomes, a bacterium's genome is just a masterpiece of efficiency. Its entire instruction manual, it's usually all right there in one single circular loop of DNA. It's lean, it's mean, and it's built for one thing above all, adapting on the fly. Okay, and this brings us to a really key concept for pretty much all life. You've got the genotype versus the phenotype. The genotype is the plan, right? The raw DNA code. Think of it like an architect's blueprint. The phenotype, on the other hand, is the actual building you construct from that blueprint. It's what the bacterium looks like and what it can do, its shape, what it eats, and this is the important part, whether or not it can survive an antibiotic attack. So here's where it gets really interesting. That blueprint, it's not set in stone, not at all. It can be changed, and that's the real secret to how bacteria evolve so incredibly fast. So let's dig into how those changes actually happen. The first way is through something we call mutation. Honestly, the best way to think about this is like a simple typo. When a bacterium copies its DNA, which it does constantly and at incredible speeds, sometimes little mistakes are made. Most of the time, these typos don't matter, but every once in a while, a random error accidentally creates a brand new superpower. And these typos can be pretty minor, like changing just one letter in a word. That might not change the sentence's meaning all that much. But sometimes they're huge, like deleting a letter at the beginning of a sentence. That can make the entire rest of the sentence complete gibberish. Either way, for the bacterium, the consequences can be massive. But here's where the story takes a really fascinating turn. Because random mistakes, that's just one part of the equation. See, bacteria don't just sit around and wait for some lucky accident to happen. They have this incredibly active, dynamic way to evolve. They share. And this gets us to what you could call the bacterial social network. The technical term is horizontal gene transfer. And what it means is that bacteria don't just pass genes down to their kids, vertically. They pass them directly to their neighbors, horizontally. It is the absolute key to their rapid adaptation and honestly, one of the biggest engines of evolution on our planet.
They've got three main ways of doing this. First, there's transformation. Think of it like genetic scavenging. A bacterium can literally just soak up pieces of DNA that are floating around from its dead neighbors. Then there's transduction, which is like a viral delivery service. A virus accidentally grabs some DNA from one bacterium and then injects it into another one. And finally, you have conjugation, which is the most direct method of all. It's like a bacterial handshake, where one bacterium builds a tiny bridge to another and physically passes a piece of DNA across.
So what is it that they're sharing in these exchanges? Well, very often it's these amazing little things called plasmids. If the main genome is like a computer's operating system, then plasmids are like genetic USB drives. They're these small, separate circles of DNA that carry extra software, specialty apps that maybe aren't needed for day-to-day life, but can be total lifesavers in a pinch. And these genetic apps are super specialized. You've got F plasmids, which basically give a bacterium the software it needs to do that handshake, to conjugate. You've got others that make toxins to wipe out the competition. And then you've got the R plasmids. The R stands for resistance, and this is it. This is how it happens. One bacterium develops resistance, packages that gene onto an R plasmid, and then starts sharing that USB drive with all its neighbors. And just like that, a superbug is born. And just to make things even more wild, bacteria also have these things called transposons, or jumping genes. These are little bits of DNA that can literally cut and paste themselves from one spot to another,
say from a plasmid into the main genome. It's just another way that useful traits, like antibiotic resistance, can spread like wildfire through a population.
So, for billions of years, bacteria had this incredibly sophisticated system for editing and sharing DNA. And for the last few decades, we humans have been learning how to hack that exact same system for our own benefit.
A totally essential tool in our hacking kit is PCR, the polymerase chain reaction. The absolute best way to think of it is as a genetic photocopier. Let's say you have just one tiny, microscopic fragment of DNA. From a crime scene, a virus, whatever, PCR lets you make millions, even billions of identical copies in just a few hours.
The process itself sounds complicated, but it's actually really elegant. You just find the little bit of DNA you're interested in, you run it through these cycles of heating and cooling with some special enzymes that we borrowed from bacteria, and boom, suddenly you have enough of that DNA to actually see and analyze. This tech is at the heart of almost all modern biology.
So, using tools like PCR and everything we've learned about plasmids, we can now do something that was once pure science fiction: genetic engineering. This is where we stop just reading the genetic code and we start actively writing it. We can insert genes, delete them, edit them, basically get an organism to do what we want it to do.
And the applications? They are just staggering. We've literally turned bacteria into tiny factories that pump out life-saving medicines like insulin. We use these genetic tricks to develop new and better vaccines. In agriculture, we can engineer crops to fight off pests or grow in tough climates. We can even do microbial detective work, identifying the exact strain of bacteria in an outbreak just by looking at its unique collection of those plasmid USB drives. All of this brings us right up to the present day and to the edge of an even more powerful future. We have learned so much by studying the secret social lives of bacteria, but this story, it is far from over.
The latest chapter is being written by technologies like CRISPR-Cas9. And here's the amazing part. CRISPR is not something we invented from scratch. It is a gene editing tool that we discovered inside bacteria. It's their ancient immune system, something they evolved to fight off viruses. All we did was figure out how to borrow it, creating what is essentially a search and replace function for DNA that has a level of precision we could only dream of a few years ago.
So where does that leave us? We've gone from just watching bacteria to learning their language, and now to becoming fluent speakers of that language ourselves. As we truly begin to master this fundamental code of life, the question is no longer can we rewrite it, the real question is, what will we choose to write?